Coming up ahead in this episode of Xtalks Spotlight.
So having an objective biomarker such as EEG would be extremely helpful in facilitating the assessments where one could rely on objective biomarker and not necessarily go 100% with the subjective report of the skills.
Hello, and welcome to Xtalks Spotlight, illuminating insights from subject matter experts and industry thought leaders. I'm Sonia Hunt. In this episode, we're asking the question, does biopharma drug discovery need to evolve to include objective, longitudinal, broad-spectrum, real-world data?
It's well established that drugs for psychiatric conditions suffer some of the lowest approval rates when compared to other major therapeutic areas. This may be because many psychiatric studies have yet to leverage invaluable tools, such as cognitive function assessments and digital biomarkers, including electroencephalography or EEG. In this Xtalks Spotlight edition, I recently sat down with Dr. Snezana Milanovic, Senior Director of Translational Medicine and Early Development at Sumitomo Pharma America. Dr. Milanovic is a physician scientist and psychiatrist, and she specializes in drug development for psychosis and mood disorders. The views expressed by Dr. Milanovic during this interview are hers individually and do not necessarily reflect the views of Sumitomo Pharma America. Thank you for taking the time in this Spotlight interview, Dr. Milanovic.
Thank you, Vera, for having me. I really appreciate the opportunity to talk about our collaboration with Cumulus.
So to start us off, there are many documented challenges with the diagnosis and treatment of neuropsychiatric disorders that clinicians face in clinical practice. As a psychiatrist who is also a translational scientist focused on developing new treatments, how do you find those challenges impact clinical research?
Thank you for the question. And this is something that we consistently think about when we design trial. Global assessments that we're using nowadays may not accurately diagnose the patient. And also there is a challenge of the timeline and when does the patient present to the study and what are the ongoing stressors, if at all, in patient's life. Additionally, we have challenges such as multiple medications that patients are receiving in the trials that might confound the overall picture. And also, very often we do not have the access to the full electronic records from the outpatient setting, which would provide us with really good insights on the time course of the disease, response to the medications, and also any complexities of comorbid symptoms. So to recapitulate, we do need better and more objective assessments. Scales, though, they're helpful and they're grant as an overall orientation for clinical practice. And obviously, we've been running trials for many, many years with the scales. There is a pressing need to move beyond the scales and try to identify reliable biomarkers of both the traits that would give us a clues about biological footprint of a disorder, but also the state biomarker, which would help us understand the course of illness and possible fluctuations and help us design better drugs.
And as you know, paper-based assessments are still widely used in clinical practice today. However, they have limitations. From your perspective, how can EEG-derived digital biomarkers address these challenges and advance clinical research?
So there is a lot of complexity in interaction between a patient who has a psychiatric diagnosis and any treater, including the principal investigators and the study staff that interact with them during the study. So we are very well trained about these interactions, and one of them is, of course, the possible placebo effect and how the alliance between the subject and clinician, whether that's in a clinical trial or in outpatient practice, may contribute to placebo effect when patient, for example, wants to please the treater, and also may confound the type of the report that patient is disclosing, and in particular the severity. So often so in clinical setting, we see patients who tend to underreport and also overreport their symptoms. So in a clinical setting, we tend to solve this problem by forming therapeutic alliance, which may, of course, confound the possible outcome with a placebo effect. But in the clinical setting, this is unfortunately not possible given relatively shortness of the trial and lack of access to all of the history to the patient and all of the electronic documentation. So having an objective biomarker such as EEG would be extremely helpful in facilitating the assessments where one could rely on objective biomarker and not necessarily go 100% with the subjective report of the skills. Let me just step back and give you an example which might be more palpable for our viewers. So let's say you have a patient who has cancer, and let's say that's a stage 3 cancer, which comes with complications such as infection and various subjective symptoms. So we can assess this patient by asking him or her about general symptoms of fatigue, of ability to concentrate, of ability to conduct day-to-day tasks and have a good grip or approximate grip on how patient is feeling, versus we can order the tests where we can assess what laboratory tests are abnormal. Is the white blood cell count too high? Did the thrombocytes drop? So we can assess what is the actual risk for the patient. And we usually couple these objective assessments with subjective, which help us come up with a reasonable picture and the best practice guidelines for the patient. We are lacking those objective assessments in psychiatry. So when patient comes and reports all of these various symptoms in regard to the typical scales, we do not have that measurement that we would love to have and correlate with subjective report and link it to long-term outcomes. So EEG, in my opinion, would be well suited if proven to be reliable for making these psychiatric assessments more objective.
Now let's talk about Sumitomo's work with Cumulus Neuroscience. Sumitomo recently engaged Cumulus on a phase 1 clinical study, and a poster was presented at the European College of Neuropsychopharmacology meeting in October 2023. What were the aims of the study and some of the key findings?
We were very happy with this collaboration. Because the task was very challenging. So visual evoked potentials are used often in the academic setting, and that gives a luxury of prolonged assessments and longitudinal follow-up of patients who are in any studies. And these prolonged assessments vary in visual evoked potential from 60 to 90 minutes. And this is, of course, not possible within the setting of a study trial where we're trying to build up drugs and assess them. So one of the major objectives was, can we shorten this standard visual evoked potential paradigm, and can we do it in such a way that it's no longer than 10 to 15 minutes maximum, and receive reliable data in the sense that data can be assessed and the signal quality would be comparable to the signal quality of a regular paradigm. And that was task number 1. And task number 2 was, well, if we can do that in the setting of a pilot study, can we do that in the real life, real clinical trial life, so to say, setting on the inpatient unit, where we would need to train also the staff who never had exposure to something like this, and also to make sure that there's reliability when these assessments are repeated, and that we choose right patients who have a good signal. So those were 2 major tasks. which were successfully implemented. And we're now working on assessing the analysis and how can we actually improve this task even further. And I would like also to highlight, which I should have probably said at the very beginning, that this task is very important to us because we want to have an estimate of the plasticity of neurons, and that estimate is done by using LTP-like paradigms, like in auditory evoked potential and visual evoked potentials, which were shown not only in human studies, but in animal studies to reliably induce plasticity, and in particular in animal studies to reliably induce LTP. So we are hoping that this will bring us a step closer to assessing some of our candidate compounds, which are shown to modulate LTP in animal models. And of course, we would love to translate that into the clinical setting.
And finally, to bring things back to our main question, do you think biopharma drug discovery needs to evolve to include objective, longitudinal, broad spectrum, real-world data?
Absolutely. So if we just step back and think about psychiatric drug development, for example, last 20 years or last 30 or 40 years, we have not bring to the market for the longest period of time a drug of a new class. There was recently schizophrenia drug that was brought as a new mechanism of action, but that happened only this year. There was like a huge gap of multiple decades, over 50 years of producing and bringing to market drugs that, for example, in schizophrenia realm are more or less like a Me Too type of mechanism with somewhat differentiation on the side effects, but even though that's not complete, we know that certain class of antipsychotics suffer from typical side effects that we would like to avoid in a new class development. So similar situation is in development for the other disorders in psychiatry and diagnostic groups. So we are hoping if we would use either visual evoked potentials or auditory evoked potentials that shown reliability measurements in animals that we can translate into humans, that we can integrate this EEG signals into digital biomarkers in an overall platform that Cumulus offers, and do really comprehensive phenotyping. I would use the example that's most similar again to the oncology. We all look after the oncology, where of course doctors and patients have a possibility to pull out the cancer cells in vitro dish and figure out how they respond to various drugs and come up with a personalized drug regimens. Similarly, we would love to collect all of these data through comprehensive digital and EEG platform and find a way to define heterogeneity of our patients for the beginning. It might not be immediately personalized, at least subgroup of patients, and have a better understanding of these biomarkers change over time and response to treatment to enable us better decision-making in clinical trials. And hopefully that might, that's our long-term passion and dream, might trickle down into also clinicians' practice to help guide their decision-making process about putting patients on certain medications and how to titrate certain medications.
Well, thank you very much, Dr. Milanovic, for speaking with us today. We greatly appreciate your time and insights.
Thank you very much, Vera. It was a distinct pleasure to highlight our collaboration, and we hope it will be inspirational for people viewing this Q&A. Thank you.
We look forward to learning more about Cumulus Neuroscience's work in neuropsychiatric clinical research. Thank you all for joining us for this Xtalks Spotlight feature. We hope you enjoyed the discussion.